Hello and welcome, dear students. Today we are talking about applications of crop biotechnology. The main objectives of today's lecture are to understand various biotechnological interventions for improvement of crop plants and to highlight the role of plant tissue culture and transgenic technology in agriculture. Biotechnology is defined as a set of tools that uses living organisms or parts of organisms to make or modify a product, improve plants, trees or animals, or develop microorganisms for specific uses. Broadly speaking, biotechnology is any technique that uses living organisms or substances from these organisms to make or modify a product for a practical purpose. Modern crop biotechnology includes a range of tools that scientists employ to understand and manipulate the genetic makeup of crop plants for use in the production or processing of agricultural products. Modern biotechnology has two well-established branches, number one tissue culture and number two genetic engineering. Plant tissue culture methods are either used to increase the efficiency of crop plants or to achieve specific objectives like micropropagation, embryo rescue, haploid production, etc., which are not possible through the conventional breeding methods. Recombinant DNA technology, also known as genetic engineering, refers to the modification of an organism's genetic makeup using transgenesis, in which DNA from one organism or cell is transferred to another without sexual reproduction. The ability to manipulate individual genes and to transfer genes between species that would not readily interbreed is what distinguishes genetic engineering from traditional plant breeding. Recombinant DNA technologies, besides generating information on gene sequences and function, allows the identification of specific chromosomal regions carrying genes contributing to traits of economic interest. Plants that have been genetically modified using recombinant DNA technology by introducing a gene from either the same or a different species are known as transgenic plants, and the specific genes transferred are known as a transgene. Transgenic plants resistant to biotic and abiotic stresses, improved nutritional quality, modified seed storage proteins, etc. have already been developed. Recent developments in plant molecular biology and genomics is revolutionizing our understanding of the ways genes, cells, organisms and ecosystems function and is opening new horizons for understanding of plant genomes and even the possibility of modifying them. Now, let us discuss the applications of plant tissue culture and genetic engineering in crop improvement. First of all, we will discuss the applications of plant tissue culture. Number one application is biochemical production. Plants are the source of large variety of primary and secondary metabolites, but secondary metabolites are of special interest since they have diverse biological activities such as antimicrobial, antibiotic, insecticidal, hormonal and pharmaceutical properties. Many secondary metabolites are also used as flavors, fragrances, etc. The term secondary metabolite is applied to all those compounds which are not directly involved in primary metabolic processes. These include alkaloids, terpenoids, quinones, steroids, phenylpropanoids, etc. Plants produce these compounds naturally but in very low amounts. Cultured plant cells have been used to improve the biochemical yield considerably. For example, chikungunya, an antiseptic drug, is produced on a commercial scale from cell cultures of Lithospermum erythrorhizon. Similarly, berberine with antibacterial and anti-inflammatory activities is produced on large scale from Coptis japonica cell cultures, whereas Taxol, which is used against breast and ovarian cancers, is produced from cell cultures of Taxus species. A number of other compounds like ajmaline, rosmarinic acid, ginseng, etc. are also produced on commercial scale. all these biochemicals produced from plant cell cultures are comparable to those derived from natural products in their chemical, biochemical and biological properties. Number second application is micropropagation. Generally, vegetative reproduction is achieved by cutting, budding, grafting, etc. to produce genetically similar progeny. However, rapid colonial propagation of plants can also be achieved by tissue culture techniques such as proliferation of axillary buds, induction of adventitious buds and somatic embryogenesis. This is called micropropagation. Micropropagation is being utilized for the commercial production of several ornamental and fruit trees such as Dalbergia shishu, Eucalyptus, Begonia, Phlox, Chrysanthemum, etc. It is also being used for the large-scale multiplication of rare and endangered medicinal plants like Aconitum chasmanthum, Aza directa indica, Rheum emodii, etc. The chief advantage of micropropagation is extremely high multiplication rates and is therefore an important technique for rapid multiplication of rare genotypes and of plants having low multiplication rates such as forest trees. Number third application is production of disease-free plants. Some crop plants are highly infected by viruses and fungi. Severe infection can cause death of the entire crop or can reduce fertility and yield. These infections continue to the next generation through seeds and asexual propagules. It has been found that shoot apical meristem are generally free from infections. In tissue culture technique, meristem culture is therefore often used to recover disease-free plants from the infected plants. Disease-free plants have been produced in dahlia, potato, sugarcane and several other crop plants. Next, fourth application is production of haploid plants and homozygous lines. Anther or pollen culture technique have been used for the production of haploid plants in several crops like rice, wheat, barley, etc. These haploid plants are of considerable significance in basic and applied research. For example, haploid plants can be used for studying the expression of recessive genes, isolation of mutants, and for determining the basic chromosome number and ploidy level of a species, etc. Through colchicine treatment, the haploid plants produced can be converted into double haploids or homozygous diploid plants, also known as pure lines. It generally takes 6 to 8 years to produce a pure line through conventional breeding methods. Such pure lines can be produced within 2 to 3 years through anther culture. Homozygous diploid lines can also be produced through anther or pollen culture from F1 plants of an interspecific cross followed by chromosome doubling through colchicine treatment. This technique is called hybrid sorting. Generally, it takes 6 to 10 years to select an ideal hybrid from the hybridization program followed by continuous selfing of the hybrid to make it completely homozygous. Hybrid sorting through tissue culture can reduce this time to 3 to 4 years only. Hybrid sorting has been achieved in wheat, barley, tobacco, etc. In these crops, several varieties have been developed through anther culture. Another application of tissue culture is embryo culture. Embryo culture technique is being routinely used in some crop improvement programs for recovery of interspecific hybrids, overcoming dormancy and shortening of breeding cycle. In some distant crosses, interspecific or intergeneric, embryo fails to develop due to embryo degeneration. In such cases, embryo culture is being used to recover hybrid plants, a technique called embryo rescue. Recovery of haploids through embryo rescue has been successfully achieved in several crosses such as Hordeum vulgare into Scale cereal, Triticum A. estivum into Agropyron repens, etc. In case of Hordeum vulgare and Hordeum bulbosum crosses and Triticum A. estivum and Hordeum vulgare crosses, haploid embryos are produced. Embryo culture is then used to recover the haploid plants from such crosses. In orchids, seeds lack any stored food and the embryos are virtually naked. Thus, orchids are difficult to propagate through seeds. However, embryo culture technique has been successfully used to overcome this problem. In some fruit trees like prunus, Texas, iris, etc., embryos require a period of after ripening varying from 1 to several years before germination. This period of dormancy can be effectively eliminated by embryo culture. Another important application of tissue culture is somatic hybrids and hybrids. Protoplast culture is an important in vitro technique for production of somatic hybrids and hybrids. A somatic hybrid is produced by fusing protoplasts of two different species, whereas a hybrid contains nuclear genome of one parent and cytoplasm of two different parents. Protoplast fusion is used for the production of somatic hybrids and hybrids between sexually incompatible species or between two non-flowering or sterile lines for creation of superior hybrids and as well as for transfer of desirable characters from a related species into a crop species. Examples where somatic hybrids have been achieved are Nicotiana plumbagineifolia and Atropa belladonna crosses, Brassica oleracea and Brassica comprestis crosses, Brassica napus and Brassica nigra crosses, etc. Now, let us discuss some applications of genetic engineering. In crop improvement programs, the most important application of genetic engineering has been the transfer of genes from unrelated organisms into crop plants for specific requirements. Such genes are called transgenes, and the plants containing transgenes are known as transgenic plants. The transgenic crop plants are also known as genetically modified crops or GM crops. Some of the most important applications and achievements in transgenic technology are discussed here. Number 1, herbicide resistance. Herbicides normally affect processes like photosynthesis, biosynthesis of essential amino acids, and other processes. Millions of dollars are spent worldwide on use of herbicides for controlling weeds. However, these herbicides not only kill the weeds but cause damage to the crop plants also. Development of transgenic plants resistant to certain herbicides was the first major achievement from genetic engineering in plants. Transgenic plants resistant to several herbicides like glyphosate, sulfonylurea, etc. have been successfully developed and are in commercial cultivation. Some successful examples are: number 1, the transgenic petunia plants resistant to herbicide glyphosate were developed by transfer of a gene EPSPS known as 5-enol pyruvate shikimate 3-phosphate synthase involved in the biosynthesis of aromatic amino acids like tyrosine, phenylalanine and tryptophan. Resistance was achieved through overproduction of this enzyme. Number 2, herbicides like sulfonylurea and imidazoline target the enzyme acetolactate synthase, which is involved in the biosynthesis of amino acids like leucine, isoleucine and valine. Transgenic tobacco plants expressing a mutant form of acetolactate synthase gene has been produced, which is resistant to sulfonylurea. Number 3 example is the enzyme glutathione S-transferase detoxifies the herbicide atrazine. Another enzyme, phosphinothrycin acetyltransferase, detoxifies the herbicide phosphinothrycin, while as the enzyme nitrilase detoxifies the herbicide bromoxynil. By introducing these herbicide-resistant genes, transgenic plants have been produced in tomato, potato, soybean, cotton, etc. Number 2 application of genetic engineering is insect resistance. Insects are believed to cause around 10% loss of the total crop production, and approximately $7.8 billion are spent annually on insecticides. Further, these insecticides are non-degradable and cause pollution problems besides killing beneficial pollinators and other insects. Major classes of insects that cause damage belong to Lepidoptera, which includes butterflies and moths, Diptera, which includes flies and mosquitoes, Orthoptera, which includes grasshoppers and crickets, Homoptera, which includes aphids, and Coleoptera, which includes beetles. Most widely used insect-resistant genes are the Bt genes, isolated from different strains of gram-positive soil bacterium Bacillus thuringiensis. The Bt genes produce an endotoxin called crystal protein or cryprotein. When this protein is ingested by the insect, it is dissolved in the midgut lumen, releasing an active toxin protein. The active form of this protein then binds to specific high affinity receptors in the insect midgut cell membranes, leading to the formation of pores that disturb the cellular osmotic balance. Within minutes, the midgut cells are paralyzed and causing death of the insect larvae. A number of Bt strains are now known, each producing a specific cryprotein against a specific insect belonging to Lepidoptera, Diptera, and Coleoptera, etc. The transgenic crop plants carrying Bt genes are popularly called as Bt crops, for example, Bt cotton, Bt brinjal, Bt maize, Bt tomato, etc. Bt technology is now a multi-billion-dollar industry in the world. Transgenic maize, potato, and cotton expressing Bt genes were among the first transgenic plants released in USA. Another major application of plant genetic engineering is abiotic citrus, leading to drought and salt resistance or tolerance. Abiotic citruses like drought and salt citruses are the major contributors of food insecurity and poverty in developing countries. Drought alone affects more than 70 million hectares of rice growing land worldwide. Resistance against abiotic citruses like drought, salinity, and coal, which all lead to dehydration problems in plants, is achieved in plants by the accumulation of osmotically active, low molecular weight, non-toxic compounds collectively called osmoprotectants. These compounds help in water uptake and water retention and also protect macromolecules from damage caused by these citruses. Some important osmoprotectants include mannitol, proline, glycine, betaine, etc. Some of the important drought and salt tolerant genes that have been transferred in different crop plants include choline dehydrogenase, glutamine synthetase, betaine aldehyde dehydrogenase, mannitol 1-phosphate dehydrogenase, choline oxidase, etc. Transgenic plants carrying any of these enzymes are capable of synthesizing osmoprotectants, which in turn confer tolerance to the plant against particular citrus. Transgenic plants where tolerance has been achieved include tobacco, rice, alfalfa, tomato, etc. Another major application of genetic engineering is improvement of seed protein quality. Cereal seed proteins are deficient in lysine, whereas pulse seeds are deficient in sulfur-containing amino acids like methionine and tryptophan. The absence of these essential amino acids limits the nutritional value of cereals like rice and wheat and pulses like pea, soybean, etc. for man. Another major application is golden rice. Vitamin A deficiency causes 500,000 children to become partially or totally blind each year. Traditional breeding methods have been unsuccessful in producing crops containing a high vitamin A concentration. Moreover, the vitamin A, that is the beta carotene, is generally present in fruits to which the poor population does not have much access. Efforts have been made to transfer genes responsible for vitamin A synthesis into rice. 3 genes providing phytoene synthase, phytoene desaturase, and zeta-carotene desaturase and lycopene cyclase were transferred into rice by agrobacterium-mediated transformation. The resulting transgenic rice, popularly called golden rice, contains good quantity of beta-carotene which gives golden color to rice grains. Efforts are also being made to transfer such genes to mustard to produce transgenic golden mustard. Since major part of world population, particularly developing countries, have rice as their staple food, it is expected that golden rice in future will address the problem of vitamin A deficiency. Another important application is eradication of iron deficiency. Iron deficiency causes anemia in pregnant women and young children. About 400 million women of childbearing age suffer due to iron deficiency. Anemia has been identified as a contributing factor in over 20% of maternal deaths after giving birth to a child in Asia and Africa. Another application is antisense RNA technology. Antisense RNA is complementary to mRNA of a particular gene. During gene expression, the DNA strand of a gene oriented as 3-5- direction in relation to its promoter is transcribed. That is, mRNA is produced from the antisense strand of the gene. In antisense RNA technology, the sense strand of the gene, which is in 5-3- direction, is inverted and reinserted into 3-5- direction in relation to its promoter. In tomato, the enzyme polygalactoronase degrades pectin, which is the major component of fruit cell wall. This leads to the early softening of fruits and a deterioration of fruit quality. By antisense RNA technology, transgenic tomato has been produced in which the activity of polygalactoronase enzyme is blocked. This has led to slow fruit ripening and increased shelf life of tomato fruits. Such tomatoes were approved for marketing in USA under the name Flavor Saver. The Flavor Saver tomatoes have improved flavor and total soluble solids, in addition to the enhanced shelf life. Another example is viral resistance. The antisense RNA technology has been used to produce transgenic crop plants resistant to viruses. In this approach, the sense strand of specific viral coat protein genes and other genes have been transferred to crop plants. The antisense RNA produced by transgenic plants blocks the expression of viral coat protein genes, thus conferring resistance in crop plants against that virus. For example, in potato, resistance against potato leaf roll virus, and in tomato, resistance against tomato golden mosaic virus has been achieved. Another important application of plant genetic engineering is edible vaccines. Vaccines are available for many of the diseases that cause widespread deaths in developing countries, but they are often expensive to use as well as to produce. As a result, the vaccines often do not reach those that need them most. In conventional approach, the inactivated form of a pathogen is used as a vaccine. In case of many pathogens, the gene encoding a critical antigen has been isolated and expressed in bacteria or animals, and the recombinant protein so produced is used as a vaccine. Such vaccines are called recombinant vaccines. In transgenic technology, the crop plants are being developed which will produce the vaccine in their fruits. Such fruits are called edible vaccines. In this strategy, the gene encoding an active antigen protein is isolated from the pathogen and after suitable modifications is transferred into the transgenic crop plant where the antigen is expressed in the plant. Banana has been used as a model plant for this technology as its fruits are consumed raw. Potato plant has also been targeted for the production of edible vaccines. Another application is biochemical production. Transgenic crop plants are being developed to produce novel biochemicals. Some successful examples are: number 1, hirudin. The first successful example is the production of thrombin inhibitor protein called hirudin. The recombinant hirudin is now produced in Brassica napus at a commercial scale. Another example is polyhydroxybutyrate (PHB), which is used for synthesis of biodegradable plastics with thermoplastic properties. At present, polyhydroxybutyrate are produced by bacterial fermentation, so the cost of biodegradable plastics is quite high compared to synthetic plastic. Attempts are being made to produce polyhydroxybutyrate in transgenic plants to reduce the cost. Genes encoding two enzymes, acetyl coenzyme reductase and PHB synthase, and the gene PHBA, which are involved in polyhydroxybutyrate synthesis from acetyl coenzyme, have been transferred from the bacterium Ralstonia eutrophica and expressed in Arabidopsis thaliana. Similarly, transgenic poplar is being prepared, which will express polyhydroxybutyrate in their leaves. The leaves can then be collected and used for PHB extraction and preparation of biodegradable plastic. Another example is cyclodextrins. Cyclodextrins are oligosaccharides which form inclusion complexes with a wide variety of organic molecules and have application in drug delivery, flavor and odor enhancement, and removal of undesired compounds. These compounds are normally produced by bacterial fermentation of maize starch. A bacterial gene, cyclodextrin glycosyl transferase from Klebsiella pneumonia, has been transferred into potato. The transgenic potato expresses the gene in tubers but in low levels. Efforts are being made to produce cyclodextrins in transgenic plants at a large scale. That is all about today's lecture. In this lecture, we have discussed various applications of crop biotechnology, such as micropropagation for mass propagation of rare endangered plants, embryo rescue for recovery of hybrids, transgenic plants for biotic and abiotic citruses, golden rice, edible vaccines, etc. Thank you very much.